Goedemiddag iedereen hier aanwezig en diegenen die via de livestream meekijken. Ik heet iedereen welkom en ik open hier bij deze academische zitting, waarin mevrouw Merel van der Thiel haar academisch proefschrift in het openbaar zal verdedigen. De hele ceremonie zal in het Engels plaatsvinden op uitdrukkelijk verzoek van de promovenda. En ik schakel dus nu over naar Engels. The opposition will start after the candidate has given her layman's presentation of 15 minutes about. And I ask you to start your presentation now and wish you the coming hour lots of success and inspiration.
Dear Pro-Rector, dear members of the Defense Committee, dear family, friends and colleagues, both online and on-site. In the upcoming 15 minutes, I will summarize the research that I've committed over the last few years. But let's talk about us first. Humans eat food to get nutrients, which gives us energy to be active, and then we produce waste products. The cells in the brain work exactly the same. They get oxygen and use nutrients. This gets delivered by the blood vessels. Then they use this oxygen to be active, which leads to brain function. Likewise, when cells are active, they produce waste products, which they then deposit in the fluid that surrounds them. For many years, it remained unclear how these waste products were then cleared from the brain, until 2012, when a waste clearance system was rediscovered. So let's zoom into the brain. The brain is surrounded by fluid which is called cerebral spinal fluid. So it kind of floats around in this fluid. Then if we zoom in a little bit more, here you can see the blood vessels that deliver oxygen and nutrients to the brain cells. They dive into the brain. And surrounding these blood vessels are these pipes which are called perivascular spaces. The cerebral spinal fluid from the surface of the brain then dives into the brain through these pipes. This is similar to a sewage system, where clean water gets delivered to our houses through pipes. So then if we zoom in even more, deep in the brain, the fluid of the perivascular spaces is exchanged with the fluid between the cells, where the waste products were deposited. And this fluid is called the interstitial fluid. It picks up the waste products and it flushes them out of the brain again. Like the sewage system picks up our waste products. However, in people with certain diseases, this waste clearance system might not function properly. The waste products are not properly cleared, which then leads to an agglomeration of these products. For instance, amyloid beta and tau in Alzheimer's disease, which could lead to cognitive decline, and then that could ultimately lead to dementia. Cognitive decline is reduced function of the brain. For instance, global cognition, so overall brain function. But this could also be more specific, like executive functioning, for instance, reduced planning, or the processing of the information, how quickly that happens in the brain, or memory problems, for instance. A reduction of one of these functions is called cognitive decline, and we can measure this using neuropsychological tests. We can measure the brain's waste clearance system with a technique called MRI, magnetic resonance imaging. MRI allows to take pictures of the brain, so we can see what the brain looks like. For instance, like this picture, which is taken like this. We can also use specialized MRI to look at the function of the brain. But during my PhD, I specialized in other advanced MRI methods, which enabled us to look at the fluid as part of the waste clearance system. So this is what I worked on the first 2 chapters of my thesis. I conducted 2 literature reviews investigating different MRI techniques that were already out there that image different parts of the waste clearance system. So the cerebral spinal fluid, 1, 2, the perivascular spaces, and 3, the interstitial fluid, and in various diseases. So there's different MRI techniques for different parts of the brain waste clearance system. And I also used this in my thesis. So let's zoom into the perivascular spaces a little bit more. As mentioned, perivascular spaces were the pipes of this system. They completely surround the blood vessels, which you can see here in red, and the perivascular spaces in blue. They then dive into the brain, and deep in the brain, the fluid in the perivascular spaces is exchanged with the fluid in between the cells, the interstitial fluid. However, when waste clearance is reduced, the fluid can accumulate, which causes these perivascular spaces to enlarge. This could be because of different mechanisms. For instance, by clogging. You can imagine that if a hose gets clogged, the water gets stuck behind it, and there's this ballooning effect. It's a little bit similar in these perivascular spaces. Or if the waste products get stuck to the side of the perivascular spaces, which then block the exit from the fluid in the perivascular spaces to the fluid between the cells, which leads to an accumulation of water and enlargement of the perivascular spaces. So this enlargement could be because of multiple reasons, and these reasons can also interact with each other. Here you can see an example of perivascular spaces in the brain that surround the blood vessels. If you would take an image like that, the tubular perivascular spaces can be seen as little dots. And on MRI that is sensitive to water, you can see these little dots here. If you count these little dots, this gives you an indication of how the waste clearance system of the brain functions. So the number of perivascular spaces is considered to be a proxy of waste clearance system functioning, since it shows how many perivascular spaces are enlarged due to the presence of waste products. So we use this proxy of waste clearance of the brain to investigate the relationship between the waste clearance of the brain and that of the eye. So by putting a paper strip in the eye of people, we could collect the proteins or waste products from their tears, specifically tau, which was one of the waste products that's one of the waste products that's involved in Alzheimer's disease. So we found that in people with more perivascular spaces, as can be seen by MRI, so a reduced waste clearance system of the brain, these people also have more tau in their tears, which could indicate a reduced clearance of these proteins from the eye. So thereby, we were one of the first in humans to connect the eye and the brain's waste clearance system. So let's go back to the system of the brain. So the waste clearance system, as I mentioned, works through fluid, picking up waste products and flushing them out of the brain. But fluid doesn't move by itself. It needs specific forces to push the water, for instance, from the cerebral spinal fluid into the pipes of the brain, something that drives it. For instance, the heart. The heart pumps the blood into the brain. This goes through blood vessels, which you can see here in red, which are surrounded by the perivascular spaces in blue. The blood vessels will expand when the extra blood from the heart pumps through them. And this mechanical movement of the blood vessel wall through the cardiac pulse has an effect on waste clearance, as it pushes forward the fluid through the pipes into the brain. This happens in the very small blood vessels of the brain. So ultra-high field MRI at 7 Tesla allows us to really zoom into the brain to look at these small vessels. Thereby, we can also measure the blood flow using a specific technique which is called phase contrast MRI. This could be done, for instance, in the lenticulostriate arteries, which are small vessels in the brain that bring oxygen and nutrients to a brain region which is called the basal ganglia. And the basal ganglia relates to a lot of cognitive functions, for instance, memory, information processing speed, and executive functioning. Using this phase contrast MRI technique, previous studies have shown that in patients with Alzheimer's disease, the vessels are found to be stiffer. However, this has mainly been researched in large blood vessels of the brain, because without the 7 Tesla, it is a bit challenging to really zoom into these small structures and look into these. So in healthy vessels, the heart pumps blood through the vessel, which then gets dampened to the compliance of the vessel wall. However, if vessels are stiffer, this blood still gets pumped through, but it doesn't get dampened as much, which then leads to a lot more energy being deposited more downstream of the vasculature. So by looking at how the blood flows, we can say something about how stiff the arteries are. Using phase contrast MRI on 7 Tesla, we found that in people with smaller, stiffer arteries have more perivascular spaces on MRI. So it could be that this increased energy that does not get dampened by the vasculature will end up in the smaller vessels, which then damages the vascular wall, which then damages the tissue around it and leads to enlarged perivascular spaces. In another chapter, we looked at how these stiffer small vessels have an effect on the functioning of the brain, also called cognition. So we looked at global cognition, executive functioning, and memory. We both investigated this in patients from the memory clinic, so people that we presume to have Alzheimer's disease or pre-stages of Alzheimer's disease, and in healthy elderly people. So in the patients, we found that stiffer arteries relate to reduced cognition. However, we did not find this in the healthy elderly people. So it seems that specific mechanisms only for people who have Alzheimer's disease pathology, so amyloid and tau waste products in the brain, their stiffer arteries are related to reduced cognition, so less memory functioning and less executive functioning. Thereby, we show that there's different things involved in how well the brain functions in healthy people and in patients with probable Alzheimer's disease. Now I will talk about the third part of the waste clearance system, the interstitial fluid. We can measure the interstitial fluid with an advanced diffusion MRI technique, which is called intravoxel incoherent motion. Here you can see the brain of two people. Both of these people have an hippocampus, which you can see here in purple. The hippocampus is the memory area of the brain. And when brain cells die, this region becomes smaller, as you can see, for instance, by the person on the right. Here you can see the vasculature of the brains of both of these people. If there is damage of the vasculature, this could also have an effect on the brain. For instance, the white matter hyperintensities, which is a specific kind of vascular damage, which you can see schematically on the right. Now let's imagine that we will use IVIM to take a picture of the brains of these two people. We will take a picture like this, as indicated by the green dotted line. Here you can see the IVIM images that were taken from these example people. You can see that on the right, there is more blue and green present, meaning that there is more interstitial fluid in the brain, compared to the person on the left, who has less green and less blue, so less interstitial fluid in the brain. So we found that in people with more shrinkage of the hippocampus, so the memory region, there was more interstitial fluid in the brain. And likewise, in people with more vascular damage, there was also an increased interstitial fluid volume. Additionally, we found that in people that had more shrinkage of the hippocampus and more vascular damage, not only did they have more fluid in the brain, but this fluid was also less able to move. It was more restricted. And this could be because of the presence of more waste products, which then blocks the movement of this fluid. So to summarize, we talked about the waste clearance system, with number 1 being the cerebral spinal fluid, number 2 being the perivascular spaces, and number 3 being the interstitial fluid. We discussed how different MRI techniques can provide information on different parts of this waste clearance system in relation to Alzheimer's disease, and how, in my thesis, I applied and optimized some of these techniques. For instance, to look at the perivascular spaces, the second part of the waste clearance system. We talked about how stiffer arteries relate to more enlarged perivascular spaces, and how stiffer arteries relate to a reduced cognitive function or brain function. However, it could also be that because there are stiffer arteries that lead to enlarged perivascular spaces, these enlarged perivascular spaces could lead to a reduction in cognitive functioning. However, this is something for future research to look into. Furthermore, we discussed an MRI technique to look at the third part of the waste clearance system, the interstitial fluid, which we imaged using IVIM. We discussed how in people with more vascular damage and shrinkage of the memory area, there is more fluid between the cells. But this is also more restricted due to the presence of waste products. I would like to thank you very much for your attention, and now I would like to give the word back to the pro-rector.
Thank you very much for this well-presented talk, which gives a lot of insight, at least to me, about the contents of your booklet. Thank you for that, and you stayed very well inside the time allotted for your presentation. we have not more time left for the questioning and the opposition than foreseen, which means that we can start now right away with the opposition, the quality or the presentation by Professor Visser. And I can also announce you that all the members of the Manuscript Assessment Committee are present today, which is a compliment to the supervisors, because that is not always the case these days. So I will not present them each separately. The first 4 opponents are all members of the assessment committee. First, Professor Visser, he's Professor of Molecular Epidemiology of the Alzheimer's disease at this university. Professor Visser.
I would like to ask you some question about your thesis. First, I would like to congratulate you with the result. It's very clearly written, beautiful pictures, and also I would, this extends also to your supervisors. So I would like to have some questions about page 22, in which you give an outline of the vascular space and surrounding tissues. And I have a number of questions. So the perivascular space it's part of the clearance system, but there are other clearance systems in the brain as well. And the most well-known is the blood-brain barrier, the blood-brain barrier in which amyloid and other proteins are exchanged actively. How do you think those different mechanisms interact with each other?
Highly esteemed opponent, thank you very much for your question and for your kind words. Indeed, there's many different waste clearance mechanisms in the brain, and the glymphatic system, or the waste clearance system that I presented today, only accounts for approximately 30% of the clearance, and the blood-brain barrier is about 70%, so that plays a very strong role as well. Considering that the blood-brain barrier is also localized very closely to these perivascular spaces, the perivascular spaces kind of surround the blood vessels, there is a lot of interaction, I would say. I presume that, for instance, if there is leakage of the blood-brain barrier, which then would have an effect on the blood-brain barrier clearance as well, this could lead to enlargement of the perivascular spaces. So in that sense, by having reduced clearance of the blood-brain barrier due to certain processes, this could also have an effect on the reduced waste clearance of the system that I presented today, due to enlargement of the perivascular spaces, which could then have an influence on the fluid flow through the perivascular spaces, which could then also lead to reduced waste clearance through this system.
Yeah, thanks. That's a nice answer. So when you have a broader view on what is happening in the brain. So then, when I look at the picture, I see kind of a dopperte in the middle of that. So these are microglia cells. So what role do they have here in the perivascular space and the clearance?
So there has been some research looking at the role of the immune and inflammation in the perivascular spaces. So there have been certain immune cells that have been discovered in the perivascular spaces. For instance, they're called like perivascular macrophages. So in that sense, I do think that it has an effect on inflammation and the immune response, which the microglia play an important role in.
Yes, and they may also play a role in Alzheimer's disease because of CAA. So there is macrophages are seen around those vessels and try to remove the amyloid, which gives bleeding. So it's a very relevant observation to include it here as well. So the final question on this topic. So part of the clearance goes through the veins. So but there are no muscles or whatsoever in it to produce or get the fluid out. So how is that organized? In this outline.
If I understood correctly, you mean through the veins?
Yes. So you have the artery with the pulsations, but the veins are connected, as you described in your book, between the arteries and the vein. So how do you think that is working?
So this is actually a thing that is quite controversial in the waste clearance literature. So there's two systems that are proposed, which is one where the waste clearance, the waste products are removed from the brain through the smooth muscle cells, which is called the intramural periarterial drainage theory. And the other theory is the glymphatic theory, which is kind of an interchanged word with the waste clearance. And this is supposedly through the perivenous spaces. So there is still quite some controversy on how this happens, and also particularly on how subsequently this drainage from the perivenous spaces would work. I presume that it is more a gradient that kind of leads to this CSF and then subsequently drains it to the meningeal vessels and lymph nodes. But how this process works specifically, it is still a bit difficult to say.
Okay, so more research is needed here. Okay. Then a question about chapter 8 on the eye correlations in the PVS. So you correlated to the tau in the tears. The nice thing is of the retina is that you can see the blood vessels. So can you see any abnormalities that support perivascular spaces in the eye?
I think that is a very interesting question, and it is something that we have been trying to get more information on the retinal vessels. I think that in the eye, this system works a little bit differently. We were still trying to figure out how this works exactly, but from the literature, it says that the fluid inside of the eye then is transported through the space that surrounds the optic nerve. So it goes through the optic nerve and then through the subarachnoid space that surrounds the optic nerve. So that is kind of what the literature is currently stating about how this waste clearance system works. I think you can kind of call the space surrounding the optic nerve a perivascular space in a functional sense of it, because it transports the fluid back to the brain.
But you can see the arteries, so it's, you know, there are all kind of different forms when they are damaged in the brain. So, and that correlates with my meta-lesions. But okay, it's good to look at that in more detail later. On the final question, because the time is almost. So, okay, it's a stelling. I read it if it's. Okay, so you said, okay, the waste clearance system is currently out of way, but MRI provides proxies for that. But it's still an indirect measure. So which kind of modality would you like to test further in to get a better understanding of the waste clearance? So, yeah.
Yeah, I think that is quite. Oh, sorry, that is quite a challenge to investigate this in humans. There have been multiple techniques that can be applied. I think MRI is a very good technique to investigate this because of its sensitivity to water, and this is mainly a system that is driven by water, and because of the small scale that you can visualize. You get a very high resolution.
And without MRI, are there any possibilities there?
Well, I think there might be possibilities with PET imaging, for instance, but that might be a bit more challenging to look at the dynamic maps, but maybe there would be some possibilities there.
And proteins, like aquaporin 4, and there's a whole bunch of proteins related to damage clearance of waste. Well, yeah.
Try to give a final answer.
It's impossible, so it's okay, thank you.
Okay.
Okay, you are satisfied. Thank you, Professor Visser. Then we go to Professor Hanseel. He is professor in behavioral neurology at Université Catholique de Louvain, Belgium, Catholic University of Leuven. It is your floor, is it your?
Thank you. Dear candidate, thank you very much for your very nice book and a very clear presentation you just gave us about an important and often overlooked topic on Alzheimer's disease. I'm a behavioral neurologist. I see patients with Alzheimer's disease, and all the techniques you mentioned are not really present yet in the clinic. And one of the questions that I most often get is not what can I do about Alzheimer's disease, but first, is it Alzheimer's disease? And your thesis convincingly showed differences in the clearance system between patients with Alzheimer's disease and controls. But when we see patients at the memory clinic, often they do have something. It might be Alzheimer's disease, it might be something else. They may not be healthy. And there are many studies, besides what you have done, on other diseases showing impairment in the clearance system, like in multiple sclerosis. You touch a little bit about vascular dementia. So the glymphatic system, the clearance system, may be altered in Alzheimer's disease and in other conditions. Do you think that all the parameters that you show are able to distinguish between clearance system alterations in Alzheimer's disease from these other conditions? And what is the evidence that it is specific for Alzheimer's disease?
Highly esteemed opponent, thank you very much for your question and for your kind words. I think you touch upon a very important point there, is that for my thesis we specifically looked at Alzheimer's disease. However, this waste clearance system can be applied or is important for many proteinopathies, and it's very difficult to separate that based on these MRI metrics. For instance, we can say that there's more waste products or more toxic proteins in the brain because we see alterations in the diffusivity, but it's difficult to say which ones these are. So I think currently, or maybe actually probably never with these specific MRI techniques, it is very difficult to say which proteins they are, which would be needed to differentiate between a clinical diagnosis. However, hopefully it would be able to distinguish whether there is a presence of waste products or not. So in that sense, it could have an added value in the clinic.
To go on this line, on chapter 4, you have a very informative graphic that you haven't shown today, showing hippocampal volume, so the structure important for memory, and white matter hyperintensities, which is accurately described as a measure of vascular impairment. On this graphic, we clearly see that some individuals have pretty normal hippocampal volume, but very high level of white matter hyperintensities. So basically, they probably have vascular trouble. How is the diffusion metrics in those individuals compared to individuals with low hippocampal volume, so that they have probably more memory impairment and hippocampal syndrome, or individuals that are controlled that do not show any of these characteristics? Do you see differences, or are they as impaired on the diffusion metrics as you? So do you see a different contribution of white matter hyperintensities to the diffusion than the contribution of hippocampal volume?
So in this specific work, we didn't look particularly at the group differences between the clinical diagnoses. The participants that you're referring to are mainly participants with vascular cognitive impairment, which is also shown by the graph. So we didn't look particularly at the differences between the clinical diagnoses. However, considering these participants have more vascular damage and less neurodegenerative damage, to just say it bluntly, we did find relationships with white matter hyperintensities and a reduced diffusivity of the interstitial fluid and increased interstitial fluid volume. So in that sense, I think that both of these pathological processes have an effect on the interstitial fluid dynamics.
And are you aware in the literature, I know you haven't touched on this, but of studies looking at amyloid and tau PET biomarkers, like in the CSF or in PET, and showing different effects in different type of population on the waste clearance system? Or is that to be done?
I think That is something that is actually very interesting to look at. This vascular system is still relatively new, so the literature is growing and growing all the time. What we also have seen, for instance, in chapter 3, where we looked at vascular and neurodegenerative diseases in the interstitial fluid relationship, is that both in vascular and in neurodegenerative diseases with different proteinopathies, there seems to be alterations in this interstitial fluid, which is important for the waste clearance system. So I presume that it might be involved in many of these different diseases, but which specific ones is something that is actually still ongoing. I think there's a lot of work coming out every day on that, so that would be something we have to wait on.
Part of the slowdown might be that you are using very specific sequences and very specific machine. You have access to a 7 Tesla MRI that is not available everywhere. We do not have access to that MRI in my university. What do you think is doable with current clinical MRI for individuals that already have MRIs? That is very frequent, the classical 3 Tesla MRI. We have diffusion sequences. What part of your work is applicable on these MRI that have already been acquired?
I think there is actually a large part of this work that could be done on the 3 Tesla. So although we have also attempted to do IVIM at 7 Tesla, we are doing IVIM at 7 Tesla, which is not in my thesis at the moment. This work has been done on a 3 Tesla, so it can be applied on clinical scanners as well. Considering the phase contrast MRI for these smaller vessels, there has also been work in Utrecht where they investigated whether it was possible to do it also on the 3 Tesla. And I think the main difference with the technique that they use there is that they take the average of multiple small vessels and really look at the one large vessel. So if you would only look at one large vessel, the differences between the 3 and the 7 Tesla seem to be less strong based on the work that they have done. So there could be possibilities that maybe with a bit of alteration, we can use this phase contrast technique to look at the small vessels on 3 Tesla as well. But since this is also still quite a new technique, we would need to really optimize the sequences and the analysis techniques to make this feasible and comparable to the 7 Tesla.
Thank you. Do you have time for one last question, or am I out of time?
No, your time is up, actually. Yes. I must be crude. Sorry.
No problem. Thank you very much.
Thank you, Professor Hanseeuw. Then the opposition will be contained by Professor Robroek. He's a professor in brain connectivity and computation at his university. Today he's acting also as the secretary of this board.
Thank you, Pro-Rector. Dear candidate, my congratulations on your thesis. It's a very beautiful work that combines clinical problems with very high-end technology. I'm a methodologist myself, as I think you know, and I like the diffusion MRI technique a lot, and my questions to you focus on that. It actually follows a bit on the question of Professor Hanseeuw. He basically asked you to imagine applying some of your more advanced techniques to the clinic. My question is actually a bit of the opposite. My question is reasoning from the maximum we could do before we start translating. So you explained beautifully in your presentation and in your introductory chapter that there are multiple diffusion components that we can distinguish with diffusion MRI. Almost classically in IVM, we already have a slow parenchymal diffusion and a fast microvascular diffusion. Recently, you and others have focused on an intermediate component, which might actually have 2 different causes, also a parenchymal one or a PVS one. So there's here now 3 to 4 different clinically interesting components. What would the ideal, without limits of technology, money, or time, MRI sequence and also MRI system look like to give you a maximum chance of distinguishing those components and the changes in them? How would that look now in your experience after having written your thesis and done your work?
Highly esteemed opponent, thank you very much for your question and for your kind words. That is an interesting question. I think something that we particularly struggle with, so to say, using IVIM in the setting of the waste clearance system is that it is currently not possible to differentiate between the fluid inside of the perivascular spaces and in the interstitial fluid. And considering that if perivascular spaces are not enlarged, they are still there, this could still have an influence on the diffusion signal, and it could be present in many of the voxels. So you would always have that partial voluming effect there. For the waste clearance imaging, I think it would be therefore very interesting if there would be a possibility to separate the two. So there is some work already from the group in San Francisco who's looking at the different properties, trying to see whether we can distinguish the perivascular fluid from, well, in this case, the parenchymal diffusivity by using the properties that it might be anisotropic diffusion because it's a tubular structure. Mm-hmm. But that would mean that if you would like to do this while also looking at the interstitial fluid, you would need to have a lot of low B values in a lot of directions to be able to separate those two components. But I think that would be a very interesting direction to go into because the interstitial fluid diffusion is commonly thought to be more isotropic. So in that sense, maybe we could use those properties to separate them.
Okay, so that would amount to a lot of low B values in quite a number of directions, so possibly a problem with time there, but we might accelerate it. And concerning hardware, would you, if you were to do that in the future, do that on a 7T or a 3T, a system with a big gradient or a small gradient?
I think a system with a big gradient would be very nice. The problem with those very small B values is that you need quite a big gradient to be able to get the small B values. I'm saying the wrong way around. But, so
you need to distinguish them at least, right? You would need a big gradient for big B values, but even if you do small ones, you do 5, 10, 15, the gradient actually needs to make a difference between them, because we sometimes build our gradients to go to 7,000 and then they can't make a difference between 5 and 10 anymore. I guess that would be important.
Yeah, I think for IVIM that would particularly be important to have those loop B values so we can separate the microvascular component, although we assume that this interstitial fluid component is like relatively higher B values, like around 200 for instance. But to be able to really separate those two, you would also need the lower B values. So the smaller gradient, sorry, I just turned it around.
Yeah, thank you. Is there time for a small follow-up question, Director?
Small question.
Okay.
Short answer.
I would like to relate that to the beautiful study in chapter 4 with the memory clinic patients. It's been referred to before, where you found that vascular damage led to a higher fraction of the intermediate component and a lower diffusivity of that. Would that improved sequence that we just talked about help to improve detecting that and interpreting that physiologically, and how?
I think that it would definitely be very interesting. It would definitely give very interesting insights on that. I think that potentially the increased interstitial fluid related to the vascular damage could be more because of the enlargement of the perivascular spaces. We have looked at this as well to see whether the MRI visible perivascular spaces were related. That didn't seem the case, but that could be because maybe there's more enlarged perivascular spaces that are not visible structurally yet. So I believe that that might have a larger influence on the vasculature, while the deposition of these waste products and neurodegeneration, which could then lead to cell damage and more interstitial fluid, those would be more processes related to the neurodegeneration, so the hippocampal volume results we found.
All right, thank you very much. I give the word back to you.
Thank you, Dr. Professor Robok. Then the opposition will be continued by Professor Zinger. She is Professor in Digital Image Analysis for Healthcare at Eindhoven University of Technology. Professor Zinger.
Dear candidate, thank you very much for the insightful talk today and for your beautiful book. I have a couple of questions, of course, and the first one is a bit related to what you have already discussed with Professor Visser previously, namely your thesis focuses on MRI insights into cerebral clearance system. MRI is a large and expensive machine. Scanning patients takes also some effort. So my question is, maybe you wondered about alternative ways, doesn't necessarily mean any imaging at all, to get to assess this clearance system in a different manner.
Highly esteemed opponent, thank you very much for your kind words and for your question. As previously discussed, I think that MRI provides a very good technique imaging-wise because you can really see regional differences which could be relevant for this waste clearance system. However, there could be maybe other techniques, like something that Professor Visser is working on a lot is, for instance, the CSF through a lumbar puncture. You can look at the different proteins in the cerebral spinal fluid and get an indication of how many waste products are floating around in the CSF. But it's difficult then to distinguish how these waste products got there and which process is then not working properly. For instance, the clearance through the blood-brain barrier. That is very difficult to distinguish whether it's the perivascular clearance system or the blood-brain barrier clearance system. But that could be an opportunity to potentially look further into that.
Thank you. In the first chapter of your thesis, you state that there are 3 possible drivers of the brain clearance system: respiration, blood pulsatility, and vasomotion. Then we write that this thesis focuses on blood pulsatility. If you would choose another hypothesis, for example, respiration, how would this change your research?
So I think this would mainly change the part of the waste clearance system that I would focus on. So there is a lot of research showing, for instance, that respiration is a large driver of the cerebral spinal fluid. So through the thoracic pressure movements, it pushes up the CSF and then circulates around. As far as I have seen in the literature, the respiration seems to have a smaller effect on the perivascular fluid, for instance. However, if there is a reduced cerebral spinal fluid flow, this could also have an effect on the perivascular movement. So I think that if I would focus more on the respiration, this would probably shift my focus more towards the cerebral spinal fluid part of the waste clearance system, which with the current techniques in my thesis, I haven't focused on as much.
Clear, thank you. Then I have a question on the early stage quantification of perivascular space enlargement. This is also the topic you studied. But from your book, I understood that it's only possible with a 7 Tesla MRI. And the question I have is, what are the advantages of this early detection for patient treatment?
So indeed, with the high spatial resolution that we can achieve with the 7 Tesla, we can see these enlarged perivascular spaces at an earlier stage. Because the ones that we see with MRI are not the only ones that are enlarged. It's more a gradual process. So with the 7 Tesla, with a higher spatial resolution, you can detect them in an earlier stage. However, that doesn't mean that with the 3 Tesla you cannot detect them. It might be at a later stage, or it would be maybe a longer acquisition to reach the same spatial resolution, depending on which one you are using. So I think in that sense, it would be also possible to do this with the 3 Tesla. The perivascular space counting is actually used in the clinic as a marker of cerebral small vessel disease as well. So it has been implicated already.
And if you would use 3 Tesla, would you need maybe some smarter image analysis technique, or is it just not acquired at the moment? There is no hope to recover the same information?
I think that would be very interesting to look at. if you could use some sort of AI or deep learning to look at this to see whether you can get the same results at the 3 Tesla as the 7 Tesla. But I presume that then you would still need the 7 Tesla images to train your model. So you would kind of go back to the original point.
Okay, let's not start about deep learning. So maybe I have one more question to ask. I was wondering about your assumption on the causality into your studies, in the sense that you investigate cerebral waste clearance system and the pathology such as Alzheimer's disease or cognitive decline. What do you assume about causality between the clearance system and the sickness? Is the sickness causing the pathology in the clearance, or the other way around? And what is the influence of this assumption to clinical applicability of your results?
Yeah, I think this is always a bit challenging to say which comes first. However, I believe that our results in the chapter looking at the pulsatility with the cognitive decline, where we only found a relationship between the stiffer arteries and reduced cognitive function in individuals that we presume to already have Alzheimer's disease pathology, that would mean that when people have these waste products, these stiffer arteries then lead to more waste products and further cognitive decline. But I think it's really an interaction, and it's very difficult to say, because this vasculature can then also have an effect on waste clearance, and this waste clearance can have an effect on the vasculature. So in that sense, it is very difficult to separate. And I think this might also be different in different patients.
Thank you very much. These were my questions, Mr. Prorector.
Thank you, Professor Singer. Then the opposition will be continued by Dr. Gijs. She is Assistant Professor of Molecular Ophthalmology at this university and University Hospital. Dr. Gijs.
Thank you, Mr. Prorector. Dear candidate, first I also want to congratulate you on your work, and especially your creativity, which I noticed while working together, but also while reading your thesis. My first burning question is, of course, about Chapter 8, about the eyes, and where you found these associations between the perivascular spaces and intraocular pressure and tau levels in tear fluid. Now, my first question is about this tau protein in tear fluid. Where do you think that it comes from, and how does it end up in tear fluid?
Esteemed opponent, thank you very much for your question and for your kind words. I believe that this is still a bit of a topic to be discovered, so to say, where the tears exactly come from. And I think that the tau proteins in the tears are no different, that it is quite difficult to say where they come from. It could be, for instance, coming from the blood, which is then subsequently transferred into the tears, so to say. Or it could be maybe because the axons that end up in the eye could have a degenerative effect, which then leads to an accumulation of tau, which then would end up in the tears. Which one I believe that it is difficult to say. I actually don't know yet, but I think that there's a lot to be discovered there, and that would also give us more clarity on the results that we have found.
Yes, that's indeed true. And the two hypotheses that you give, they cannot really explain why you see then a correlation with the increased perivascular spaces, because blood is not really a component of that, I would say, and the axons neither. So there must be something else that we don't know yet. You also found this association with intraocular pressure and the perivascular spaces. Can you say something about intracranial or intracerebral pressure and the relation with the clearance system?
Yes, so in this chapter, we argue that because of an increased intraocular pressure, that would also be related to more perivascular spaces, decreased pressure, sorry. However, probably this is more an interplay between the two, so the intraocular pressure and the intracerebral pressure. So there has been some literature showing that the balance between these two is responsible for this clearance of the waste products of the eye, which would also make sense in a way if the fluid needs to move from the eye to the brain and there's a higher pressure in the brain than there is in the eye. That would be quite challenging. So I think it's really a balance between the two, but that is actually something that we would be very interested in to investigate in the future, how this would subsequently lead to reduced waste clearance of both the eye and the brain.
Yeah. And the relation or the correlations between intraocular pressure and perivascular spaces, you found them for the ipsilateral eyes or hemispheres, but not the contralateral. Why do you think that is so?
Yeah, so that was actually something that we were very surprised by, considering that the optic nerve crosses at the level of the optic chiasm. So most of the time there's a relationship with the contralateral hemisphere. However, I believe because we're talking about fluid flow, that the fluid from the eye, then it doesn't cross. The pressure kind of still goes ipsilaterally backwards. So in that sense, I think that might be the reason why we found an ipsilateral relationship and not a contralateral one.
Yeah, I agree as well. And maybe one final question. You found this association in this centrum semiovale, but not in the basal ganglia. Although this chiasm, this optic chiasm, is very closely located to the basal ganglia, I guess. So why do you think that is, that there were significant findings in the centrum semiovale, but not in the basal ganglia?
So we found this particularly for the results related with tau, which is a waste product or a protein. And there has been previous work which has shown that the deposition of waste products seem to have a larger effect on the perivascular spaces in the centrum semiovale, while the basal ganglia, the perivascular spaces in the basal ganglia seem to be more related to vascular stiffness, so arteriosclerosis, for instance. So potentially, because of these different mechanisms driving the perivascular spaces, the relationship with the waste products can only be found with the centrum semiovale and not with the basal ganglia.
Okay, thank you very much. I give the word back to the prorector.
Thank you, Dr. Gijs. Then the opposition will be continued by Dr. Schner. He's a medical physicist at Maastricht University Medical Center. Dr. Schner.
Yes, thank you. Dear candidate, I would also like to congratulate you with your nice thesis. I especially enjoyed all the figures that helped me understand the complicated medical terminology that I run into sometimes as a physicist, not specialized in this field. First, I would like to go to chapter 7, where you analyzed two groups, a group of controls and a group of memory clinic patients. You analyzed these two groups separately, but even though the range of the damping factor between those two groups is comparable, you only find a correlation between the damping factor and cognition scores in the memory clinic sample. So I think this begs the question why the healthy controls are not affected. Does this mean that vascular changes that are measured by the damping factor are not a primary driver of impaired cognition, or is there something else going on?
Esteemed opponent, thank you very much for your kind words and for your question. I think that that is quite difficult to say what exactly the mechanisms are behind this. One of the reasons why we could not find a relationship in the healthy people with cognitive decline is that they have a smaller range in cognitive decline. So although the range in the damping factor might be similar, as you can also see with the scatter plots here, there's really this cloud of the healthy people, while the people from the memory clinic have a very large range in cognitive decline. So that could be one of the factors that then has an influence on that it would be more challenging to potentially find this relationship. For the undergoing processes, I think that it's difficult to say. It seems that there might be different mechanisms at play with the cognitively healthy elderly people, so that there is not something vascular, or that there's not this damping factor that then relates to cognitive decline, but there could be other factors related to aging, because it is an aging sample of healthy elderly people. In the people from the memory clinic, I think that potentially we do find this relationship because they already have Alzheimer's disease pathology potentially in the brain, which could then lead to stiffer arteries, which could then have an effect on the vasculature, and then the vasculature potentially having an effect on reduced pulsatility and thereby reduced waste clearance system through the glymphatic system, or the waste clearance system that I talked about today. So I think it could be two different mechanisms, but to really say that reduced dampening does not have an effect on healthy elderly is still quite challenging, I would say.
But is that then, continuing on this, is that then a timing effect, that they're not yet affected because they have like similar values, like a damping factor of, I believe, something like 0.4. You have that in your memory clinic sample, but you also have that in healthy controls. Would it then take more time for the healthy controls, or are they somehow protected against this lower damping factor?
I think that could be the case. So maybe because the presence of these amyloid proteins or tau proteins is not present in the healthy controls, this could be more of like an interaction effect, that in the healthy people that we presume not to have Alzheimer's disease pathology, which is a presumption because we didn't measure that in these people, that this is not the case, and it could be that this cascade, this circular relationship, which I have discussed in the discussion as well, would only start once there is already Alzheimer's disease pathology present in the brain. But that type of causal inferences are very difficult to make, especially in a cross-sectional study like we have done now.
Okay, thank you. Would you, I noticed that you're excluding patients with an MMSE score of below 20. I understand why you would do that for practical reasons, but there's a whole part of the scale where you don't have any information. If it would be possible to include such patients, what would you expect from your, what would that give, what kind of insights could that give?
Yes, indeed, it is quite challenging to include patients with an MMSE score of below 20 due to the ability to consent to the study. If this would be possible regardless, I presume to find actually that the relationship goes even further. So we have this kind of negative, actually this positive relationship, and then if you were able to also measure the lower spectrum of that, so even more reduced cognitive decline, I would also assume that that would have the same effect with the damping factor.
So it would be even more clear.
Yes, I presume so.
Okay, then I have time for another quick question. So you look at the blood pulsatility and the damping factor and the correlation between that. I was wondering, to what extent do you think the pulsatility index says something about local vessel characteristics as compared to a more extended part of the vascular system? And how does this compare to the damping factor? And does this explain why the damping factor shows a better correlation with cognitive scores?
Yeah, so indeed, the pulsatility index we have calculated in this particular work in the lenticular arteries and the middle cerebral artery. So the blood flow velocity pulsatility would give an indication of the stiffness of the vessel at that particular point where we have measured it. So for instance, in the middle cerebral artery or in the lenticular artery. The damping factor, however, is kind of a relationship between the two. So from the larger vessel, the middle cerebral artery, to the LSA. So it would say more about the whole vascular tree.
You may very briefly finalize your answer if you wish. You can also remain silent if you wish.
I think that I quickly concluded it, so it's okay.
So that must be clear to you that now the time for the defense of your thesis is up. The committee will withdraw for consultation, and I ask you and your company to remain in this hall and wait our return here. I adjourn the meeting for a few moments.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
I reopen this meeting. Normally we do the ceremony in Dutch, but we will continue in English. That's also easier for, I understand, your loved ones and maybe for the audience which is with us on the live stream. Dear promovenda, dear candidate, the committee has discussed the quality of your booklet, of your thesis, sorry, and the quality of the defense. And based on its positive verdict on both, and in view of all your examinations you already passed, it has decided to grant you the doctor's title. Your Professor Jansen is authorized to confer upon you these academic distinctions, so the floor is to your promoter.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Merel Margaretha van der Thiel, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, secretary, and the other members of the committee, and affixed with the official seal of the university. On behalf of EURON, the European Graduate School of Neuroscience, I'm also entitled to hand out the official EURON certificate. EURON is a training network of 8 universities in the Netherlands, Belgium, Germany, France, and Luxembourg. This certificate is awarded to PhD graduates that have participated in multiple courses and conferences of the EURON program, that have collaborated and worked in a foreign research group, and that have published their work in international scientific journals. Dear Dr. van der Thiel, dear Merel, on my behalf of the supervisory team, including Professor Walter Bakkes, Dr. Ine Ramakers, and myself, it is my great honor to be the first one to congratulate you on this remarkable achievement today. I would like to extend my congratulations to your partner Steph, your family and friends who have supported you throughout this journey. Your PhD story began in 2019 when you applied for a position funded by the Alzheimer Nederland Foundation. We were seeking for someone to work on glymphatic dysfunction and cognitive impairment in a memory clinic. This was a collaboration between the Department of Radiology and Nuclear Medicine and the Alzheimer Center Limburg. At the time, you were still in London, in the UK, having just started as clinical research officer at the Imperial College. Your impressive track record already demonstrated your ambition and international outlook. After earning a bachelor's degrees in psychology in Leiden, you pursued 2 master's degrees, one in neuroscience in Geneva and one in dementia in London. And to our great relief, you decided to leave your role in London and move to Maastricht to join us. From the very start, it was very clear we couldn't have wished for a better candidate. The project, studying the glymphatic waste system and its role in cognitive decline and eventually Alzheimer's disease, was very ambitious. Imaging the glymphatic system is notoriously complex, and we aim to leverage the ultra-high field imaging capabilities of the 7 Tesla MRI scanner. This was no small challenge, both technically and clinically. However, for you, challenges were simply opportunities. You immediately took the lead, and we, supervisors, were merely passengers, occasionally pulling the handbrake when your enthusiasm led you to full speed ahead. Your PhD was not without its hurdles. The COVID-19 pandemic caused facility closures and delays in subject recruitment. And midway through your project, the 7 Tesla scanner underwent a major upgrade, complicating combining data from before and after the upgrade. However, you never really seemed worried, at least to us. If one project encountered obstacles, you already had 10 new ideas waiting to be explored. Your ambition and drive extended well beyond the scope of your initial project. You secured funding to spend time in Boston in the lab of Laura Lewis, growing your international network. You became the student representative for the ISMRM Neurofluids workgroup, and you were even invited to contribute a review on interstitial fluid and lymphatics to JMRI, one of the leading MRI journals. For this review, you collaborated with your office mates, or should I say, you instructed your office mates how to write a review. You even managed to convince those colleagues whose expertise was outside this topic to join the fun. And that demonstrates really nicely that you're a great team player. Also, the collaboration with the Department of Ophthalmology is a very good example of that. But of course, nobody is perfect. One thing we quickly learned was your love for writing, and writing a lot. With your first review, we discovered that you were the ChatGPT avant la lettre, generating overwhelming amounts of text. My first instruction was, please cut this by at least 50%, preferably more. Despite this, the final result was well written and landed in a prestigious journal. Another flaw might be your relentless enthusiasm to do everything at the same time. You often worked on multiple abstracts for multiple conferences, traveled internationally, and to our surprise, even briefly pursued a clinical psychology training, all at the same time. We sometimes had to convince you to put aside 9 other projects and focus on one critical one. Merel, I'm very proud of you. Your talent, determination, and collaborative spirit make it clear that you have a great scientific career ahead. You have already secured your first major personal grant from Alzheimer Nederland to continue your work as postdoc in Maastricht, with another visit to Boston on the horizon. I wish you every success in the future and look forward to continuing our collaboration. So.
Dear Dr. Van Thiel, yes. It is my pleasure to congratulate you also on behalf of the university and extend these congratulations to your loved ones on the first rows of this auditorium. It was a great pleasure for me to hear you. You had already high expectations, having read your CV, which is excellent so far, and now is even better with a doctor's title. We are happy that you chose this university to conclude your academic education by getting your doctor's title, although you're not an alumnus of this university, but from now on you're highly esteemed in our papers as the one from Maastricht. Thank you for that. I'm thanking you, and I wish you, of course, a successful career. The first steps have already been set, because you are now already a postdoc, and that will mean that you will have a successful career, of course, afterwards. Not only do I thank you for that, I also thank Professor Hanseo and Professor Zinger, both from other universities, to do us the pleasure and the honor of attending to this ceremony and also taking part in the assessment of the booklet. Thank you for that. It's good to have you here. The others will not be thanked because it is their job to do this. But still, we are happy that you are all here today. Yeah, we now go to the final part of this meeting with some practical points. In a few moments, this committee will leave the room in a procession in the cortege. Then we go to the main hall in the staircase and have an official picture of the group with the new doctor. If the few people who want also to be on that picture, and then I think of loved ones and father, mother, and so on, not the rest of the audience, they should we stay a bit near our team as long as the photographer is still there. And if you wait then for a few moments before we leave in a cortege and go to the refectory where the candidate, the former candidate, the new doctor, will offer us a reception and some nice, maybe some drinks, or I don't know what you have ordered. But we thank you for that as well. Having said this, I close this academic session. Allemaal even een stoet.